welcome back to my YouTube channel, and today we discuss about the chapter molecular basis of inheritance and the topic about the structure and function of DNA, and also discuss the part of the DNA. Okay, please all see my full video and try to understand all things that I teach you. Okay, the structure of DNA. I divided the topic in two videos. That means in first video I will discuss about the part of the DNA, and in the second video I will discuss about the structure of DNA. Okay, see the first video what I discussed in this first video. Okay, the structure of DNA. In this chapter main things is that what is DNA and what is the structure. Okay, DNA are extracted from nucleic acid. That means nucleic acid is of two types. One is DNA and another is RNA. Okay, this nucleic acid is divisible into two parts. One is DNA and another part is RNA. And the introduction part we comparison between DNA and RNA. Okay, DNA presents inside the nucleus, but RNA present inside the cytoplasm, not inside the nucleus. DNA present all cells and it acts as a genetic material for all type of organism. But RNA acts as a genetic material for certain type of bacteria. That means some bacteria that present in primitively, but prokaryotic cells, some prokaryotic cells, RNA is act as genetic material. Okay, then you know that one important question that comes in the exam, that is polynucleotide. Polynucleotide. What is polynucleotide? The polymer of nucleotide. The polymer of nucleotide is called as polynucleotide, which is important question comes in the exam. Okay. Then what is polynucleotide? We discuss this polynucleotide part briefly. Okay. In the polynucleotide, we discuss briefly about sugar, phosphodiester bond, pentose sugar, that means sugar and pentose sugar both are included in this part, and nitrogenous base, nucleoside, and the last part is nucleotide. We discuss all the part: sugar, phosphodiester bond, nitrogenous base, nucleoside, and nucleotide. Briefly we discuss all those components of the DNA because these are interlinked in the topic of nucleotide, and nucleotide is completely linked up with the DNA. So that we discuss all the part briefly. In the first component or the first part of the DNA is pentose sugar. Okay, we discuss what is pentose sugar. Pentose sugar is of two types. Okay, what are they? One is ribose and another is deoxyribose. The ribose present in RNA while the deoxyribose present in DNA. Here a question is arising that why we call this structure is ribose and why we call this structure is deoxyribose? Because of the needed wise oxygen is present in this ribose structure, but the needed wise oxygen is not present in deoxyribose. Because why? Here one oxygen is not present, because here the needed oxygen is present. Here not oxygen, needed oxygen is present. So that we call this structure is deoxyribose. Here I remember the things that what is the full form of DNA? The full form of DNA is deoxyribonucleic acid. Why called deoxyribonucleic? Because deoxyribose present in DNA. But the full form of RNA is ribonucleic acid because of ribose present in RNA. Okay. The second part of the DNA is nitrogenous base. Nitrogenous base is further divisible into two parts. One is pyrimidine structure and another is pyrimidine structure. Okay. Then the pyrin and pyrimidine structure further divided into two parts. That is pyrin divisible into adenine and guanine, but pyrimidine divided into cytosine or thymine. Here a extract case is present, that is in case of RNA thymine not present, that means thymine present in DNA, but uracil present in RNA. Okay, uracil present in RNA, but thymine present in DNA. This is the main difference between DNA and RNA that comes in the exam as a short question. Most important, that is uracil present in RNA and thymine present in DNA. Most important question. These are the part of nitrogenous base, which is also a important base pair created in the structure of DNA. The third part of DNA component is nucleotide. Okay, in nucleotide here two parts is present, that is sugar is present both side, and the nitrogenous base, that is adenine and thymine or ATGC, adenine, thymine, guanine, cytosine is also present, and it was added by, or it was connected by N-glycosidic linkage, by N-glycosidic linkage, and the two nitrogenous base are connected each other by hydrogen bond. A connected with T by double hydrogen bond, but G, that means guanine, connected with cytosine by 3 hydrogen bond. Okay, here the adenine connected with the N-glycosidic linkage at 9 position, but sugar in sugar carbon is present. This carbon link with the N-glycosidic linkage at first position. It is also comes in the exam as a question. Most important question also come in the exam in this part. The fourth part of the DNA is nucleotide. Many students are confused between nucleoside and nucleotide. Don't confuse between nucleoside and nucleotide. Nucleoside means this part is called nucleoside, but when it is connected by phosphate group, when the two nucleoside, it is a nucleoside and it also a nucleoside. When this two nucleoside, I call what? Side. Two nucleosides when connected with phosphate group by two phosphodiester linkage, then the whole part is called as what? Nucleotide. Don't confuse between nucleoside and nucleotide. I tell again that when sugar connected by N-glycosidic linkage with nitrogenous base, that means ATGC, this part is called as nucleoside. When two nucleosides are connected by phosphate group by phosphodiester bond, then this whole structure is called as nucleotide. Most important question, nucleotide comes in the exam as a short note, that means 2.5 marks question. Don't forget this part. Most important. Again I tell that sugar when added with adenine by N-glycoside linkage, it is called as nucleoside. When two nucleosides are attached by phosphate group by two phosphodiester linkage, then it is called as nucleotide. Okay, these all are the part of the DNA.